So anyways, I will do that though. We will have a test analysis. We're going to look at where we made mistakes. You will reflect on what you're going to improve. You're going to have, especially those of you that didn't do well, are going to come up with a study plan on how you're going to improve what you're going to do for next quarter. As I said, the content will be covered again as we go on. Now let's talk about what life is. Because this is, what is this class? And what do we do in biology? Biology. Someone turn the lights off, please. Logy is a study of and life. So we're studying this. We're studying life, and of course, you have to. In order to study life, you had to start with atoms and molecules. I mean, there's no other way around it. So let's see if we can apply some of the knowledge that we've learned thus far. What is what's the foundation? What's something that all life has in common? What are some things that all life, some characteristics? For all life. Something we can all agree with. Very good. They all have to process energy. They have to have energy. Energy is the stuff you need to move. So life moves, right? Yeah. What's another property of life? Okay, that's true. All life has mass, but it's not something that's unique to life, right? I'm not going to put that one. It's a right answer, but I'm not going to put it. Yeah.
Some way all life is water.
All life is what?
Uses water.
Uses water. I'm going to put these out on the sides, the fringes. They're absolutely right. Has mass, uses water. This is true, but I'm looking for some other characteristics that are uniquely for life. Yeah? Anyone? Yeah? Have to consume things to survive. So I'm going to leave that with processing energy. So, consumption. Now, plants make their own energy, but they still need nitrogen and CO2, so they're still consuming. Yeah.
They all have carbon?
They're all carbon-based. Yeah, all life on this planet. So these are all correct. I'm just brainstorming here. What else? Something unique, something you'd notice. Look around you. Look at the people next to you. Think about your parents. What do you think happens with life? Or your dogs, your cats, your fish? Yeah.
DNA.
Okay.
They all die.
No, it's actually
That's
it's
true.
true.
Even fish.
Eventually, every life ends. Now there are very old organisms. There's a 10,000-year-old organism in underneath Michigan that's been alive, obviously, for a very long time.
Gene and DNA?
Yeah, it's a fungus. Say what?
Not forever.
What? Fish? There are some fish that are 100 years old.
She
Several.
said jellyfish.
Jellyfish are they think are eternal in the sense that their DNA is it's not just jellyfish. Planaria is another one, but that's a little different. There's not one jellyfish that's been around for 10,000 years, but they repair themselves and essentially eternal. Yeah, that's right. They all have DNA. But what about DNA? What is it that they're able to do because of DNA?
The gene.
Genes. Be unique.
They reproduce. Life reproduces. Life reproduces. It makes more of itself internally, right? They make more of themselves. They reproduce. So what else? What else does life do? So they reproduce. They process energy. Do all these things over here on the side are true as well.
Okay.
What else? Yeah. Go ahead. They what? They're all made of molecules. I'm going to go ahead and say that carbon-based DNA mass / molecules. All right, what else? And biomolecules, right? Specifically the 4 biomolecules, right? And macro. Let's call them macromolecules, right? We should know what those are, yeah?
They all change over time.
They do. Can we say grow from within? Why do I say from within? Because rocks grow too, right? But they grow by layering or what's called sedimentation or evaporation. So they actually can layer, rocks can grow by layering. Yeah. Say heal. Yeah.
That's rough. I didn't even think about that.
Heal injury.
What's that stuff called that? Like when you cut yourself?
Um,
That P word.
Grow from within and heal injury. Yeah, all life can do that. Yeah. Yeah. Cells is exactly right. So this is a big one. Cells. And let's, so this is actually, you know, a real big deal that all life, now, this is an argument that's going on right now and from a lot of biologists. There's this thing that a lot of you know about, and we're going to put in a little red box over here, the word in a little red box, because it's a big argument. A virus is not a cell,
So what is it?
and we'll talk about it later. No, bacteria are cells. Bacteria and viruses are two different things. I know that in everyday life, people treat them the same, like that'll kill 99% of all, 99.9% of everything, including viruses. But viruses and bacteria are not the same thing. They're essentially different. Bacteria are cells. They are definitely alive. So the question that scientists have argued for a long time, I'm not even sure if it's still settled, I'm pretty sure it's not, is it alive?
Yes. If it's spread.
Most, as far as I know, most biologists say no. Why? They're not cells. That's a big one.
But
There's some other things. They also don't process energy. They don't do much of anything. Most viruses don't do much of anything.
reproduce, right?
They don't reproduce
It just
internally.
spread.
Your cells make copies of them.
Replicate, right?
Your cells copy them. So your cells are actually the ones that are picking up the virus and making copies of the virus. They have little bits of DNA or RNA in them that tell your cell what to do. Remember we said DNA is the code that's telling the cell what to do? Well, these are like little packets of information that come and steal your cells and tell your cells to do something. But that's all they are. They're not actual cells themselves. They can't process it. And we'll look more about what processing energy means. What else does life do? What did your parents do for you?
Feed it, like,
Okay, you fed it, but that kind of goes with processing energy, yeah?
yeah.
They did reproduce to make you, that's correct. But what did they do when they reproduced? Did they just reproduce, just made a new version of you? What did they do?
Degraded.
Say it.
I said that. I said they passed off genes.
They passed on genes. They passed on hereditary information, right?
No, because everybody said they on there. Everything in there looks.
So life sends hereditary information from one generation to another. All right, to the next, and so on. So that's an important aspect of life. There's a lot, as you can tell, there could be a lot. So when we're looking at living things, these are the kinds of things we're going to, there's one that maybe you haven't thought of yet. What else does life do? All life that I can think of. They what? Say it.
Said breathe.
Breathe. Okay, that's part of processing energy, but that's a good one. But breathe has a specific connotation, like using lungs to breathe. Trees don't. They exchange gases. That's true.
Yeah.
But do you really call it breathing? I don't know.
I mean,
Life responds to the environment.
Oh, yeah, adaptive and adapt.
And that's the other big one that we haven't talked about. But yeah, that's another one that's coming in a second. So it responds to the environment. What do I mean by that? If somebody comes over, well, that's actually something that's a little different. But if somebody comes over and pokes you, what are you going to do?
Smack it.
All right, smack. You're going to do what?
I'm like, what's up?
You're going to respond,
Yeah.
right? I know, you said that. That's what I put there, yeah? Response to the environment. So if somebody pokes you, you're going to respond. If you poke a tree, will it respond?
No.
Yes, it will. It will respond. It won't get up and move. It won't slap you. A Venus flytrap will. You can stick your finger in there, it'll go snap on you real quick.
that hurt?
But, no. Well, it does if you're a fly. But what's interesting is that if you poke a tree, it'll start to release sap, right?
Oh, like
And
maple syrup.
if you cut a tree, it'll start to grow new tissue to repair itself. So it's kind of related to healing injury. You respond to the environment. Sunflowers, for instance, will follow this. It's a plant, right? It'll follow the sun across the sky. It'll actually turn as the sky moves across. As the sun moves across. Actually, it's the
So
earth
do you say
turns.
sap, you mean like maple syrup trees and stuff like that? Like you poke
Yeah,
the thing in there?
yeah. If you take a tube, that's how we get maple syrup. Is we go to a maple tree and we tap it. It's called tapping. You tap a tube into it. And it'll slowly release sap, and we collect the sap, we
You ever
boil it,
tasted
and
that?
collect, it's
Like,
delicious, yeah.
Yes, boy, it is.
Yeah.
Maple syrup without the tea.
Shh, huh? If you cut, depends how you cut it, what kind of tree it is, how good are its roots. It's a lot of good questions on that one. They will grow back. You can do a cutting. It's called a cutting. Cut a branch off and grow a whole new tree. It's a whole discussion there that we have to get a little bit deeper into biology to really understand what's going on there. All right, so we understand that's a good collection, I think, for now of what life is. So we're going to focus on this part, and all we did first quarter was try to get ready to be able to talk about this part right here. So what is a cell?
Oh.
The one big thing that people haven't mentioned that I thought was a big, yeah.
I mean, the senses count as the senses go down and down, like the five senses.
Say that again.
Like the five senses.
The five senses. Okay, so let's think about that. Seeing. Not all life like humans do, right? But not all humans have all five senses. Like some humans are blind, some humans are deaf, right? Some humans have problems feeling. But not all living things hear like we do, right? Like trees can't hear like we do, although they can sense sound. Some of them can sense sound. It's an interesting thing. They don't process, they don't have brains. Not all living things have brains, right? So that's another thing. Not all living things have nerves. Trees don't have nerves. Not all living things have bones. Jellyfish don't have bones. Right? So there's all kinds of things that are different among. So when we say characteristics of life, that's a big umbrella, right? But we have to think about stuff that all life does. And these things here, all life do. Right? Yeah.
Water.
They need water. We put that up here, but that's right. Uses water. That's good. Those are smaller ones. It's true. These are all true things. But here's another thing about all life, because we talked about inheriting information. All life adapts. All life adapts. And how does life adapt? From generation to generation. We use the word adaptation. Huh?
To the environment.
Adapt to the environment, that's correct. Now, there's two ways to adapt to the environment, right? Like you put on a coat if it's cold, or you, for instance, a rabbit, there are some species of rabbit, in the winter, they turn white. Their coat turns white. In the spring, their coat turns gray. Now that makes sense, right? They're adapting to the environment. But those are just genes turning on and off. But there's another kind of adaptation. So there's short-term adaptation, right? What's the other? And what's the long-term adaptation? Generational adaptation. That's right. It's called
Oh, so it's
evolution. You said it right. Which is what frustrates me about you not doing work, because you make these amazing statements and then. So short term, generational, so that you can adapt as an individual to the environment by changing your behavior, right? You know, so you're going through, so think about the kinds of adaptations you do. You go from one classroom to the other, one classroom, one teacher wants it one way, the other teacher wants another, so you have to adapt, right? So that's an adaptation. So those are short-term adaptations, but then you have a generational adaptation, so one generation to the next can change. Fundamentally, the population can change, and if the population changes, we call it evolution. And we're going to take a really close look at evolution. All of these we're going to take a look at, all these characteristics of life. Are we all good so far? All right. There are others, but we'll focus on these for now. What is a cell?
it's the parts that make up your body.
Okay. But what is a cell? What is it? But describe it a little better. What do you know about it?
It
Yeah.
got a nucleus, got a pre-kill.
Okay, hold on. Yeah. It's a living, okay, it is. So you got a beaker.
Contains information.
We're gonna look at some cells too in a lab soon.
And
So you have a glass or a beaker, a plate or something. How do you, come on people, focus. What am I gonna do? What am I gonna do with this? How am I gonna draw a cell? What do I do?
it's real small.
Is this a cell? It's a glass. There's water in it. There's water. Is that it?
No.
Yeah. You draw a circle. Most people draw circles. That's true. They don't have to be circles, but they could be. So you need something. If this is the environment, and can I call it outside? What do I need?
Inside.
I need an inside. How do I get an inside if I have an outside already? Someone close the door, right? Huh?
Zoom in.
No.
It's already something inside. It's warm.
Hey, we have an outside. How do we get an inside?
You gotta zoom in like real close.
No, there's an outside. What do I need to have an inside? If we're outdoors. What do we have to have in order to get indoors?
Water, air.
Way to get in.
Okay, so what you're saying to me is if we're in the forest, now listen and think for a second. You're in a forest, and there you are, and here I am. And we're out in the woods, okay? And you're telling me all I need is a door, and now I got an inside and outside, that's it? I need what? Okay, shelter, what do I need?
You need something.
I need walls. I need a separator. I don't want to call them walls because I'm trying not to use the word walls because it's a very specific word with cells. Something to close it in. Yes?
Yes, sir.
So now if I go ahead and close it in, now I have an inside and an outside. But I have to have something to close it in. Huh? I can't hear you.
She said a cell membrane.
That's right. If you said cell membrane, that is correct. Nice. So we need a cell membrane. I don't want to use cell wall because that's only found in plants, bacteria, and fungi, right? You don't have any cell walls, but you do have cell membranes. And the cell membranes are made of what? You all know it, or you should know it. Somebody said it. Phospholipids. Your cell membranes, I'm going to go ahead and draw it out here. That's right, cellulose is only in plants because now pay attention please. So you have these phospholipids, and when you have phospholipids, it turns out in cells have a bilayer. Remember we talked about this? I didn't put it on the test, but we have phospholipids. You can look at the postings if you don't have it in your notes. And I also drew it for you here on the board. And so you have in with a phospholipid, you have a hydrophobic layer and a hydrophilic layer. So if there's water out here, there's water in here. So this is inside. This is outside. This is inside. Now, if I were to draw it, somebody said it was a circle. That's right, it would be something like this, right? I would draw a circle. Somebody else earlier said there was a nucleus, there was a Golgi body, there was endoplasmic reticulum, there's ribosomes, all those, there's DNA in there. That's a cell, right? Those are cells you all learned about in middle school. But what specifically was left out in most people's education in middle school is this idea of a cell membrane. And this cell membrane is made of phospholipids. It goes all the way around the cell. It's also, there's another, it's called the plasma membrane. Which is the outermost membrane. It's actually, there's a number of membranes in eukaryotic cells, but all cells have this one separation. This one outer separation. It's called the plasma membrane. And it's not stiff. It's constantly moving. These things here, these phospholipids are spinning. They go around, spin this way. They flip. This phospholipid will flip down there, and this one will come up here. So there's constantly changing and moving. We call it a fluid mosaic. Do you know what a mosaic is? Model. Fluid mosaic model. All right, has anybody ever seen stained glass?
Yeah.
Anybody ever seen a picture? You ever see like a bunch of colored tiles where somebody put them together to form an illustration? All right, that's called a mosaic. Let me show you what it looks like on the big screen here. I can't show it to you there. These are mosaics. Right? So this is a famous one there. These are all different colored tiles that come together to form, right? You all agree they're all different colors, different shapes, different sizes. Yes? Yes. But together they form one picture. Agreed? Okay. So that's called the mosaic. So when you look at a cell membrane, it's actually made of a lot of different things. The main component is phospholipids. A hydrophobic part is where? Where's the hydrophobic part in this membrane? The center. This is all hydrophobic here. The part that's pointing towards the outside and the inside are made of what? They're what?
Hydrophilic.
They're hydrophilic. Why? Because they're pointing towards the water. So I'm doing yellow because it's like fat, right? So it's hydrophobic. But the outside's hydrophilic because inside the cell is water and outside the cell is water. But the membrane is the thing that divides them, can't dissolve in water. Otherwise, the whole thing doesn't work, right? Function and structure, think about it. This inside has to be hydrophobic because if it dissolved in water, it couldn't very well act as a separator. Do you agree?
Yes.
Could I make, for instance, if we were out in the woods again, could I make this house out of sugar? Not gingerbread, but sugar. Especially if it rains a lot. Why not?
Yes, you can. Cellulose.
No, I can make it, well, okay, very good. But I'm talking about dry eating sugar, sugar we can eat.
No.
Why not? Because as soon as it rains, what's going to happen? It's going to dissolve. So it wouldn't be a very good shelter. It wouldn't be inside and outside for very long. So a membrane has to have something that does not dissolve in water, that can hold its integrity. We don't build our houses out of cardboard because when they get wet, they turn into mush. We build it out of something that's going to resist water. So we have to have this layer that's resisting water, and that's that hydrophobic part. Then there's a hydrophilic part. We have to have a hydrophilic part because what must it do?
What you mean?
Why not just have a hydrophobic part? Why do we need the hydrophilic part? Because what?
Change that.
Because what must the cell be able to do? Let's think about it. Let's look at a bloodstream. What's in the blood? This is an artery. What's in the, you know what's in the blood, at least one thing's in the blood. The blood cells.
Oh, yeah, red and white. Oh, no.
Red, let's look at a red blood cell. There's a red blood cell. Why is it important that the outside of these cells are hydrophilic? Because the blood is mainly made of water. And if it was just hydrophobic, would it be dissolved in the water? Would it be? Could the cell float in that? It would do what? If this was a pure ball of fat, it would just sit at the top. It wouldn't be able to float around. It wouldn't be able to move around freely. But because these molecules have a hydrophilic end and a hydrophobic end, the hydrophilic end can be dissolved in water, both inside and out, and the hydrophobic part can separate the two, inside from out. And so you have this little mobile space that can move through the blood and not be dissolved. So it's actually the property, this dual property of phospholipids, this polar end and the nonpolar end, that allows you to have a cell. You're going to build some cells next week as an activity, and you're also going to look at them underneath microscopes next week.
Wait, which one's the hydrophilic and which one's the hydrophobic one?
Hydrophilic is the head, hydrophobic is the tail. That was part of your studies for the last exam. You see why it was important that you learned that. So we got the wall, that makes sense, or not a wall, but let's say a separator. So we have the separator. What else do we need? Look at your inside and outside. What do you need? You need an opening. And that's right. I mean, it sounds kind of ridiculously simple, but it's not really. So I'm going to draw this. I'm going to draw the red, the blue, and so this is going to represent. So there's, come on, focus, will you please? So there's your hydrophilic, there's your separator, right? Your membrane. There are your heads. And you're sticking out towards the water ends. I'm not even going to draw the tails. Let's just assume the tails are there. So now my question is, how do we make, why can't we just put a hole in it? Why not just put a hole in it?
Because it's got to be closed, and then it got to...
Gotta be closed.
Because you want to control it, right?
Yeah.
Don't you want to control it opening and closing? Do you just want a hole? I mean, could you just leave your window open all day long in the house?
Yes.
Why not?
I do.
Somebody will come in that you don't want to come in, right? They'll steal your stuff.
So like,
You'll get a skunk in your bed. That'll be a bad thing.
I'll be dead.
Get a rat in your toothpaste. That would just suck, wouldn't it? Go get your toothpaste, and then there's little beady red eyes looking back at you. So what are we gonna do? What are we gonna do in order to make? We need an opening, but what must we have with that opening?
A door.
It has to be a door. It has to be something that opens and closes, right?
The insulin-
So I'm gonna worry about this thing called sugar.
Oh yeah, like the
I'm
insulin.
like it's very good, very good. See, there is a reason I teach you things,
Well,
doesn't
you
I?
wasn't even at case with us.
But we talked about it.
Yeah, we totally in class. So let's draw a sugar. Just a simple hexagon. You got to get this thing through. It's dissolves in water, so it's polar. So this is polar. It's not gonna. Will it dissolve through this nonpolar area? No. So it can't get through. It can't get through the membrane. But what we do have, however, interestingly,
It's a key to the door.
well, we need a door first. So we need a door that opens and closes. We need a space. What we use is these magic things that do almost everything in your body. What's the one macromolecule does almost everything in your body? Proteins. We need a protein. And so we can make this protein that looks like this. Shh, pay attention. And I'm making up what it looks like. Just
What's that?
A protein.
there's your door. What must this end be? What this part of the protein, what must it be? It must be nonpolar. Very good.
Good job.
This is excellent. This is your I'm very happy that you said that. This whole on the outside, this all has to be nonpolar. Do you see that? Does everybody see that?
Yes.
That this part has to be nonpolar because that's the part that's in the membrane. On the outside, what must all this on the outside be? Polar. So all the amino acids, all the R groups on this side of the protein are all polar or ionic. And on this side, all of them are what? Are on this side? These all must be polar or ionic because this is water, this is water. See how the heads are all pointing to each end? Like dissolves like. There must be a way for the sugar to get in, right? Because if the sugar is going to come in, there has to be a tube, right? So the tube's going to go this way. That slotted thing is a door. I'm drawing it cut out. Do you notice that I'm drawing it cut out? Do you understand that? So this is cut out. So it's like I'm cutting an orange in half. Or I'm cutting this protein, this membrane in half. And so this part here must be nonpolar. This must be nonpolar or hydrophobic. This part here must be hydrophilic. Is it time to go?
Yes.
All right, we'll finish this tomorrow.
Hopefully.
Do this.
You have hydrophilic on the outside, hydrophilic on the inside. The inner part's also going to be hydrophilic, right? How do you get hydrophilic all the way through, though? Well, remember, this is only on the outside is it hydrophobic. On the inside, the whole thing's hydrophilic. Does that make sense?
Wait, say it again.
So here you have a bunch of amino acids all connected together. All the amino acids that make this up on the outside, they're all hydrophilic. They're all either charged or partially charged, right? Either they're polar or fully charged, and they're sticking out. All the amino acids that are charged or partially charged are sticking inside the cell. The ones that are nonpolar are stuck in the membrane. Correct? Mm-hmm. Now inside the tube, this is a tube. This is a drawing of a tube. So if I were to draw it sideways, it would look like this, right? So let me redraw this with less of a. So if I were to draw the tube, it would look more like this, right? Where the inside would be blue, and the outside, in the area where it's stuck in the membrane, would be yellow. But then this end would be all blue, right? And this end would be all blue. Does that make sense? That way you can get the protein stuck in the middle. But these are all amino acids. They're making a tunnel through the membrane. You see the tunnel?
So that's the whole membrane.
This is the membrane, and then there's a tunnel through the membrane. This is a tunnel. It's called a transport protein. It's called a channel. We call the tunnel, when we have tunnels in membranes, we often call them channels.
Transport protein. The middle is.
It's a channel. A channel is just it's like a tunnel. Okay, it's a channel is a connector. Does that make sense?
Did you say the middle is a transport protein?
The whole thing's this whole thing is transferring stuff from outside to inside. It's allowing things to move through it.
So the protein want to go inside.
The protein's stuck in the membrane. It's stuck. See the yellow part?
Yeah.
It's never going to go into the water because it's what? What's the yellow part?
Hydrophobic.
Hydrophobic. It's going to stay in the membrane. Once it's in the membrane, it's set.
So what's that square then?
This part of the protein is what? Hydrophilic. And this part of the protein is
I
hydrophilic.
mean, what's that shape that's supposed to be going?
This is sugar. It's trying to get in. But it can't fit through the membrane because sugar dissolves in water. It's polar. So it can't, it's hydrophilic. So it can't get through the membrane. It cannot get through the membrane. So it needs an opening. It needs a door. What's the door? This protein, this channel. But we don't want just sugar just rushing in whenever it feels like. We want to control it. What molecule do we have to control it? Insulin. So we have this molecule that's insulin, and I'm going to go ahead and erase a little bit here just so I can move it around. So the sugar's trying to get in. And remember, sugar has a this is glucose, so it has a kind of looks like a hexagon. It's trying to get in here. And there's a bunch of these. There's a bunch of these sugars, because when you eat a candy bar, you get that sugar in the blood. I'm not going to keep drawing them, but you get my point. There's a lot of these sugars in the blood. They all want to get in, but they can't fit through the membrane because the membrane is nonpolar here in the middle, and they're hydrophobic here in the middle, and this is hydrophilic. Okay, so they can't get through the membrane. They need a channel. This is the channel, but the door is closed. Right, the door is closed. Well, the way insulin works is a little complicated. It's much more complicated than just a lock and key. What happens is that there's another protein over here. So in this case, I drew it closed, but really I should have drawn it as open. So as long as the channel's in the membrane, sugar can get in. But the channel's not always in the membrane. The channel's not always in the membrane. So as long as you have a membrane, the channel only lasts for a short time. So what your body does is over a little bit further down, they have another protein that looks like this. And I'm totally making up the shape, but it also has a hydrophobic part and a hydrophilic part, just like the protein we just looked at. It's not a channel. It's what we call a signaling. It's a receptor. So what do you think this is going to receive? Yeah, it's another one. So when there's a lot of sugar in the blood, there's a receptor here that accepts something. What do you think it's going to accept?
The sugar.
Well, you would think, right? The sugar is going to take the insulin. So what happens is insulin is released by your pancreas, and let's do the insulin in red. The insulin binds to this protein. No, not did. Insulin. Insulin binds to the protein, and then on the inside, there's a whole list of changes, a whole bunch of chemical reactions that happen that tell your DNA, hey, make more. It tells your DNA this. It says, let me do a call out. I guess that's better. It says, make more channels. So it sends a signal. The insulin literally is a hormone that your pancreas releases. It tells your cell, in all the cells of your body except your brain. Your brain doesn't need this. Your brain has these in the membrane all the time. But insulin binds to all the other cells of your body, and sends a signal from that protein that's in the membrane down to the DNA that says, hey, make more channels. Because what makes proteins? DNA. So DNA says, okay, I'll make more sugar channels, and makes them, then sends them through a bunch of different pathways, and you get a bunch more of these channels in the membrane that lets what? Sugar out of the blood and into the cells. So when you're high blood glucose, when you have insulin, you make more of the proteins, the sugar leaves the blood and goes into the cells.
What was that called?
Goes from outside to inside. This is a receptor. It's a receptor. This is the hormone, this is the receptor.
You said
Say
allow
it
sugar
again.
to leave what?
Allows the sugar to leave out the blood and go into the cell. Yeah.
What happen if it's too much sugar going in at one time?
You get diabetes.
Wow, no, that's don't confuse it. Yeah, there's a little bit of there's a confusing statement there. So that's a great question. That's a fantastic question, and it kind of when we will do a case study on diabetes as well. So there's another receptor. This is how. Remember I said fluid mosaic, right? Mosaic, a bunch of different parts and pieces. So there's another receptor down the line here.
Oh my, I can't draw all this.
Yeah, because insulin also.
Dang, it's another one.
There's hundreds of these. This is a very complicated, your membrane's very complicated. It doesn't work any other way. It couldn't do all the things it can do. There's another hormone that your pancreas releases is called glucagon. Gluc from glucose, right? Glucagon. So when your blood glucose gets too low, that glucagon binds to a cell receptor that tells your body, hey, let's get rid of these channels, too many. So it calls out that receptor. That's another message that says too many channels. Your blood glucose is getting too low. So they get rid of the channels. They stop making them. The DNA gets shut down. Stop making them, right? These disappear out of the membrane. So now your blood glucose goes up again. So your blood glucose is constantly going up and coming down, going up and coming down, but it stays around some average number. You see how it goes? Stays around some average number. This is the number you want to stay at. This is normal. Sometimes it's a little higher, sometimes it's a little lower, but you want to stay around this over time, right? And this is how it does. You have one hormone that says, hey, get rid of the channels. The other hormone says, put the doors in. Take the doors out, put the doors in. Take the doors out, put the doors in. That's happening all the time. Every time you eat something, your pancreas is releasing. You see how complicated this is? And you see how many different ways this can get broken. So it explains how type 2 diabetes can happen in a bunch of different ways. Some could go wrong with glucagon. Some could go wrong with the receptor. What happens with people like myself who have type 2 diabetes, our receptors don't work that well anymore. So now when my pancreas releases insulin, but my receptor, its shape is changed a little bit, so it's not able to read the insulin molecule like it used to. All right, so this is just one example of many, many different kinds of proteins that we'll be talking about this year, but different channels. And we're going to do a lab. We're probably going to end up doing like 3 labs next week, so be ready for that.
That means 3 lab reports.
No, I'm not going to do that to you. We will do lab reports, but I'm not going to have you do a full lab report on every lab. It'd just be too much work. But we will be doing another lab report soon. But my point is this. How you get stuff in and out of the cell is going to be important. And this is just one example, one way. And I talked about insulin right now because we went to the medical school and we talked and we had that series on insulin. And I think it's important that you understand that this is all connected. So this insulin molecule is a protein. Glucagon is a protein. It's made by DNA in a pancreas, in an organ called the pancreas. And this insulin and this glucagon are released into the blood, and that either makes doors that allow sugar in or gets rid of the doors to take sugar out or allow sugar to build up. So just to be clear, that somebody said it right a minute ago. So let's think about this. These proteins. So I quickly sketched this out when we did it. So this is a protein that sticks out here. Maybe I should change the color, right? Maybe it'd be better if I just changed it to blue, just to make it consistent. So if I drew this in here, right? And the insulin has to be blue as well, doesn't it? And this side's blue. What part of the protein then would be hydrophobic? It would be here, this part, correct? Because it's in the membrane, it must be hydrophobic. So the amino acids that make up for the receptor protein, the amino acids that stick in the membrane for this integral membrane protein, they're both integral membrane proteins. This is a channel protein, and this is a receptor protein, a signaling protein. So in this protein over here, there's two sections as well, isn't there? There's the section here that's sticking out into the blood, right? There's the section that's sticking into the cell. And then there's a section that's in the membrane. So again, this part's sticking out into the water, so it's has amino acids that are made of hydrophilic, and perhaps I should draw the glucagon as blue as well, right? Just leave it as a black outer layer, because glucagon is free floating in the cytoplasm, in the blood, right? Rather, so it should be blue, right? And really, sugar should be blue, because it's all of it's hydrophilic. All of these molecules are hydrophilic. They're in the blood, therefore they have to be able to be dissolved in water. But the part that's stuck in the membrane, that part has to be hydrophobic. Agreed? Now, take a look at this one really quickly. This membrane, the outer portion that's facing the lipid is yellow. But there has to be a part of this membrane that's blue. Does that make sense? This part here has to be blue. The part that's touching the inside has to be blue. Why does it have to be blue? Because it has to be continuous with the outside. So it has to be hydrophilic, otherwise they wouldn't allow water through. They wouldn't allow sugar through. Does that make sense? So the amino acids that are facing this channel have to be, and remember, what does that look like? This channel is cut. So it looks like this, right? Where the tube, the inside tube, all the amino acids on the inside of the tube are all hydrophilic. That means they're either charged or polar. All the ones on the outside here and here are hydrophilic, so it means they're charged or polar. But the ones on the outside here, which are represented here in this yellow, they have to be hydrophobic. That's what sticks them in the membrane. Are we clear on this? So that's going to be on the quiz on Wednesday. So let's talk about the quiz on Wednesday. So let me write this stuff down so we have it on record. Number one, Wednesday's quiz, right? Wednesday's quiz. This is going to be pretty straightforward. Number one, you need to know what a plasma membrane is. You need to know what is it made of. What is it made of? You need to know which is and which is is that right? All right. All right, so hydrophilic, yes, I understand the eye's a little messy, my bad.
It's two L's.
He just Googled
No, it's only one.
It's only one. He just Googled it. What is that?
Although this whis is not right. That's not right. And nobody caught that as far as I know. Oh, somebody was? Okay, all right.
He thought he was up to it.
So which is hydrophobic, which is hydrophilic, right? What is it made of, the plasma membrane? You should know what the fluid mosaic model is. Right, and you should know what an integral membrane protein is. And remember, that's a protein that's embedded in the membrane. You should understand.
That's GR.
Yeah, that's GR. Okay. I'm trying to hurry up and get this done so I can actually get on to the other stuff, but okay. My bad. Integral membrane proteins, proteins, hydrophobic part. Because these integral membrane proteins have to have a hydrophilic and hydrophobic part, right? And the proteins also have a hydrophilic part. Correct? All right. Finally, you should have an understanding, and it's a basic understanding. The whole process is complicated, but I think you should have a simple understanding of, and it's all spelled out in the previous notes here, an understanding of insulin and how it works, and an understanding of glucagon and how it works, okay? All right, so understanding this, I think is a major part of the quiz on Friday. Now let's go on. Not Friday, I'm sorry, Wednesday. Today is Monday.
So we got it Wednesday.
Your quiz is on Wednesday. Now let's go on.
Talking 2 days.